Hi, today I am going to discuss about production of streptomycin. Okay, before going to the topic, let's see what is streptomycin. Streptomycin is a bacterial antibiotic drug. Okay, and it is used to treat several diseases like tuberculosis, endocarditis, brucellosis, plague, etc. Okay, this antibiotic can inhibit both gram positive and gram negative bacteria. So it is a broad spectrum antibiotic. Streptomycin antibiotic was first isolated in 1943 by Albert. It was the first aminoglycoside antibiotic. Aminoglycosides are oligosaccharide antibiotic. Next is the mechanism of streptomycin. Streptomycin is a protein synthesis inhibitor. That is, this antibiotic binds to the small 16S rRNA of the 30S subunit of bacterial ribosome and causes inhibition of the protein synthesis. Finally, it leads to the death of the organism. This is the mechanism of the streptomycin. Okay. So it is a protein inhibitor or the protein synthesis inhibitor. Okay. Structure of the streptomycin. Structure of streptomycin is complex. The commercially available streptomycin is hydrochloride of streptomycin. The structure include streptidine, dihydrostreptomycin, streptose, and N-methyl 1-glucosamine. Okay. Next is the organism used for the streptomycin production. Aminoglycoside antibiotics are produced by actinomycete species. Streptomycin is an aminoglycoside antibiotic. Okay, that is aminoglycoside antibiotic means the antibiotic that contains oligosaccharide or the sugar units. Okay, so the culture used in the streptomycin production is Streptomyces graeseus. Streptomyces graeseus is an actinomycete. Okay. So the first step in the fermentation process is the culture preparation. Okay. The inoculum, that is the Streptomyces graeseus inoculum, is introduced into the suitable media. Okay. The inoculum requires 5 to 7 days incubation at 28 degrees Celsius. Okay. About 4 to 5 percentage inoculum is added to the fermenter. This organism produces grey spores, and the spores are maintained as soil stock or as lyophilized culture. Next is the media used for the fermentation process. The media composition is given here. Soybean meal 10 gram, glucose 10 gram, peptone 5 gram, sodium chloride 5 gram, meat extract 5 gram, distilled water 1000 ml, and the pH range 7.3 to 7.5. Glucose, galactose, fructose, xylose, mannitol, maltose, lactose, or starch can be used as the substrate in the medium. Here, glucose is the best carbon source. Fats, oils, fatty acids used in the presence of glucose. The nitrogen and phosphate is obtained from soya meal. Then we can move to the fermentation process. Here the inoculum used is the Streptomyces griseus. It requires strong aeration and agitation. Here the continuous fermentation process is required to produce streptomycin. The spores can be produced on the medium and the spores can be stored under lyophilization or by soil stocks. The optimum fermentation temperature is 28 degrees Celsius and the process complete within 5 to 7 days. The pH requirement for this fermentation is 7.3 to 7.5 and the aeration rate 0.5 to 1 ppm. The yield is 1200 milligram per milliliter. There are three main steps in the fermentation process. Okay, let's see the three phases. First phase of the fermentation. In the first phase, the growth of mycelium occurs. The proteolytic activity of the Streptomyces griseus releases ammonia from the soybean meal and also carbon from the soybean meal utilized. So the release of ammonia increases the pH of the media from 7.3 to 7.5 into 8. Okay. So the pH rises due to the production of ammonia by the activity of Streptomyces. In this phase, the yield of streptomycin is low. That is, this phase requires about 2 days. Then phase 2. In the second phase, the streptomycin produced at faster rate. There is no mycelial growth. Okay. And the pH is about 7.6 to 8. The pH lowering is due to the utilization of the ammonia. Okay. There is no rise in the pH. Okay. In the first phase, the ammonia is produced. In the second phase, ammonia is utilized. So, in the first phase, pH is raised and the second phase pH lowered. Okay. Ammonia and glucose is utilized rapidly and this phase takes about 1 to 2 days. Okay. Then the final phase, that is phase 3. Okay. In the phase 3, sugar level depleted. Thereby the production of streptomycin stopped. As a result, the cell lyses and releases ammonia. The release of ammonia again increases the pH of the media. Before lysis, fermentative material is harvested for the recovery of penicillin. Okay, so these are the three phases in the fermentation process. Okay, totally we can say the fermentation process takes about 5 to 7 days. Okay. At the start of fermentation, streptomycin is fixed into the RNA molecule. Okay, RNA molecule present in the cell. Later, it is extracted into the culture medium. So, it requires recovery. Final step is the recovery process. After fermentation process, by using filtration technique, mycelium is segregated from broth. The antibiotic treated with activated carbon. Then elution from carbon with dilute acid. Okay. The eluted streptomycin antibiotic is precipitated by solvents. Then it undergo filtration and finally dried. Okay, these are the steps involved in the recovery process. I hope you understood the fermentation process. Thank you. Thank you all.